Hi, everyone. Welcome to another Science on Tap. I'm Linda Deck, the director of the Bradbury Science Museum. And you have come to Science on Tap to learn from the scientists themselves, the latest cool research being done here at Los Alamos National Laboratory. Today's topic, tackling the plastics problem with Dr. Taraka Dale. Science on Tap is a program of the Bradbury Science Museum here in Los Alamos, New Mexico, with support from the Los Alamos Creative District. Thanks so much to Lauren McDaniel, the Los Alamos Main Street Director, and the Los Alamos Creative District Curator, Jeremy Smith. In fact, here's Jeremy now to tell us more about upcoming Creative District events. Jeremy?
Thank you. Yes, my name is Jeremy Smith. I'm with the Los Alamos Creative District, and looking forward to this Science on Tap this evening. Creative District also brings you History on Tap, Nature on Tap, and Culture on Tap. Next Monday, April 26th, we have Culture on Tap, and that is with the Los Alamos County Art and Public Places Board. Can you hear me okay, Linda?
Yes.
Okay. So that's with the Los Alamos County Art and Public Places Board. I'll have the chair of the board as well as the staff liaison. We're going to talk about what does the Art and Public Places Board do, where do they get their money, how do they spend it, and how do they make those decisions on the art that you see around town, whether it's outdoors or indoors. The Art and Public Places collection has over 250 pieces in it, quite amazing, and we'll take a look at some of those virtually on that tour, and then just open it up for Q&A at the end of it, as always. And then next month we've got Nature on Tap. That's hosted with the Nature Center and Pajarito Environmental Education Center. And of course, History on Tap is a partnership with the Los Alamos Historical Society. If you want to learn more about all of the on taps, you can check out the website, creativelosalamos.com. You can also sign up for the newsletter there. I'll send out a reminder and updates for the upcoming On Tap. So join us next Monday, Culture on Tap, April 26th, also at 5:30. Once again, my name is Jeremy Smith, Los Alamos Creative District.
Jeremy, thanks so much.
Yes.
Really good to see you. And sorry if I had an odd look on my face when you were talking. It's because I was trying to close a very annoying box that was in the middle of my screen. And that reminds me, strange. So, but that reminds me to help people get the very best experience in using WebEx. You will want to go to the upper right-hand corner of your screen where it has the layout, and you're going to want to click for the full screen view. That's going to help you have the very best experience tonight on this program. All righty, so thanks for telling us what's coming up. Really fun is that this is Earth Week, and this program is part of Earth Week. So this Thursday, April 22nd, you could enjoy an Earth Day afternoon from 1 to 3 p.m. This is through the laboratory with a presentation from former laboratory director Dr. Terry Wallace on the natural wonder of northern New Mexico, followed by the Bradbury's very own Mel Strong. He is our educator here at the Bradbury, one of them. He's also an atmospheric scientist, and Mel is going to explore the perennial question, will it rain this summer? We always wonder that here in New Mexico, and we want it very much. So I look forward to hearing what Mel has to say from 2 to 3 on Thursday, the 22nd. To know how to get there and for other Earth Week activities and programs, just go ahead and visit the homepage of the museum, which is www.lanl.gov/museum. And you can find the link right there. Now, what's coming up with Science on Tap? Well, in May, on May 17th, Science on Tap will feature geophysicist Charlotte Rowe. Charlotte will be discussing the use of smart cables to explore the Earth's secrets beneath the sea. And then on May 26th, another upcoming program. I want you all to mark your calendars for this one as well. The Bradbury and Los Alamos National Laboratories Fellows will be presenting the next installment of our Frontiers in Science series. Raymond Newell will examine how quantum physics can keep the energy grid safe from cyber attacks. That is going to be a very interesting program. May 26th, that's a Wednesday. So folks, here's how Science on Tap works. If this is the first time that you've been joining us, you've done the right thing right off the bat. You've joined us because it relies on you too to be here. Please, we want you to have a good time. We have a presenter who will be sharing their work, and this presenter will give 20 minutes, maybe up to a half an hour of a discussion on the work that they're engaged in. And then it's up to you to ask questions, and that's what will keep the conversation going. And feel free when you ask a question to refer back to another question that was asked. That will simulate what it's like when we're able to be together in person. So write your questions. The important thing is, please, write your questions into the Q&A, not the chat. Write it into the Q&A and send it to all panelists. Then I will be able to see them and I will serve them up to our speaker. I'm sure we can have a lively conversation. So our presenter today, as I mentioned, is Dr. Taraka Dale. She's a biochemist at Los Alamos National Laboratory. She's part of a group that focuses on algae utilization and other microorganisms in the production of cost-effective biofuels and bioproducts. She is active on 9 separate projects, including 4 research and development consortia supported by the United States Department of Energy's Bioenergy Technology Office. She's the team leader of the biomass and biodiversity team. And if that wasn't enough, she's also currently serving as the Bioscience Division's acting deputy division leader. That's a heavy lift. That's a lot of administrivia. So congratulations to you, Taraka, for keeping up with all of that. In her spare time, Taraka enjoys spending time with her family, traveling, hiking, camping, skiing, and eating well, which I really identify with too. So, welcome, Taraka Dale.
Thanks, Linda. Thanks for having me here. So, thanks for tuning in today. I will be talking about just one of the projects that I helped to lead. It's called BOTTLE, which stands for Bio-Optimized Technologies for Keeping Thermoplastics Out of Landfills and the Environment. So it's quite a wordful and mouthful, I guess, and so we're glad to have a shorter acronym to go with it. So we all know that plastics are everywhere. Many of you will be aware that plastic is in our oceans. There's been a lot of stories out there in the popular press about all of the plastic waste that has made it into our various waterways and really specifically into the open sea. And some of these pictures, as shown here, are just really, really devastating in terms of the amount of waste that's out there. And some studies have even projected that plastics may take up as much weight in the ocean as fish are in the ocean by the year 2050. So this is a really massive, literally, problem for us across the globe. Recently, a study that may not have made it out quite as widely yet because it's just much more recent is that plastics are even in our air. And so this is not a problem just of plastics in our landfills and plastics in the ocean, but really right in our backyard in some of our most precious areas in the United States. Our national parks and other areas in the Mountain West have been found to have small microplastics and small fibers in both the in the air, and therefore landing and blowing into regions that are really precious to our country. So plastics really are everywhere. And of course, we're all familiar with the types of efforts that are out there for managing our plastics waste. We know all about being able to recycle our plastics and how we have to sort them, and how there are certain regions where you can't even recycle all of the polymers or all the different types of plastics that are out there. Maybe a community can recycle 1s and 2s, but they can't necessarily take, number 5s, for example, which is probably, something that people are quite familiar with. And of course, other types of waste such as, glass and composting and et cetera can be recycled or can be utilized in other ways besides just going to the dump. But the challenge is that, in fact, even when we do have these approaches for recycling, only a small fraction of our plastics make it through the recycling process. And when they do, they're converted into something that's of less value than they started with. So there's not a lot of incentive to try to kind of reclaim that plastic waste or the carbon in those products. So some of you might be wondering why Los Alamos National Laboratory is focused on this particular problem. But it turns out that renewable products are actually really a part of LANL's mission. And so there are a number of grand challenges that have been recently identified by the laboratory as part of their National Security Life Sciences Initiative. And renewable products and making alternates to petroleum products fall both into our environmental grand challenge as well as our bioeconomy grand challenge. And so working in this space really meets multiple national security missions in energy, materials, and climate. And this is again a problem of real magnitude. In fact, 6% of the world's fossil fuel consumption is used just to make polymers. And so this is as much as the amount of fossil fuels that goes into the global aviation sector, and is a real fraction of the amount of petroleum that we use every day. So not only does Los Alamos and other national laboratories care about this problem, and we know consumers care about this problem, but companies are really starting to take this to heart as well. And so if you've had a bottle of Coke in the last handful of years, you might have noticed that they have a symbol on their bottle that says plant bottle. And so they're taking very seriously the idea of using different types of polymers or polymers made from biomass in their products or in their packaging. And the Ellen MacArthur Foundation has also put out an initiative wherein they have a new vision for creating a circular economy for plastic, and they have gotten a number of big name companies to sign on to their vision for making more renewable products for their packaging specifically. And so companies understand that plastics play a major part of people's way of life. I certainly understand how convenient and embedded plastics are in everything we do every single day, and that's not likely to change really soon. But we also know, and companies also know, that environmental sustainability is important to their customers, and it's important as good global citizens.
Taraka, I really appreciate it that companies are wanting to do their part. Personally, I know you can hardly unpack anything, take it out of its packaging, and it doesn't have plastic in it. And I always check to see, does it have a symbol to tell me what kind of plastic it is that I can recycle? So many of them don't. And I hate to have to throw that away. And so many things are those number 5s that we can't recycle here in Los Alamos. So if there was a way to get the companies on board with being able to do something with it that comes back to them, right? That it's an economic boon for them to do it. We would think so much more fondly of those companies as well, right?
Right. Yeah. I think that's correct. And so, Yep. So, and we can talk a little bit more about the company's interest in this particular project too, maybe when we get to the end. The project that I'm here to speak with you about today is called Bottle. We are just one of 10 institutions that are a part of this project. The project is led by the National Renewable Energy Laboratory, which is another national lab up in Colorado. And there are altogether 5 national labs and 5 academic institutions that take part in this consortium that's funded by the Department of Energy. And if you're interested in knowing where to find us or to learn a little bit more about us, it's pretty simple. It's just bottle.org. We're continually updating our website, but there's definitely some information out there for you guys to check us out further. So we have really 2 primary goals within Bottle. So Bottle's vision is to enable a circular plastics economy. The idea of a circular economy, especially around renewable products, might not be new to some of you. It's definitely a concept that's out there. But just to explain a little bit more about what this means. So what I've been referring to in the last handful of slides is really the concept of a linear plastics economy for the most part. So, you know, you mine your petroleum, you take it out of the ground, you take it to the refinery or to your other factories, and you make the products that you're using every day. And then we dispose of those. And as Linda mentioned, some of those, you know, we can actually recycle, but a lot of them end up in the dump. And so then they're going to the landfill. Some of that is leaking out into our environment, either because it never made it to the dump in the first place or because of just the way trash tends to move around. And some of it may be burned in order to try to get rid of it. And then, of course, then we have particles going into the atmosphere. So one goal for Bottle is to develop technologies that help us degrade these existing plastics. And not only do we want to just degrade them, but we want to then turn them into products that are more valuable than what they started with. And this is a concept that's called upcycling. And so typically, if you take a product and even if you do what we normally call recycling, you're not actually recycling in the sense that you're turning it right back into something that's just as valuable as we started. It actually is almost always turned into something less valuable. And that does not incentivize then companies to want to retrieve back their own waste because they're not able to turn it into something that they could resell at as good of a price or higher price. And so for our vision of a circular plastics economy, we want to design and to invent new building blocks that allow us to make plastics that work just as well as the plastics we have today, but these polymers would be different in that they are actually much easier to recycle. And so in this concept, the plastics are turned back into their building blocks that then can be flipped back in to the polymers. And so instead of making something that's of less value in this particular cycle, you're making the exact same thing over and over again. Excuse me. And so we want to design new plastics that are easier to degrade or recycle in the future. And there's a lot of ways that one could go about this. And as I mentioned, this is a big project. And so we are actually tackling both the idea of deconstruction, which is degrading those existing plastics, and the upcycling and the redesign, making those plastics for the future in a couple of different ways. And so this cartoon just shows the idea of having various types of plastic waste. And then different members of the team are looking at different ways to break down that plastic into small building blocks. And then other members of the team are looking at ways of turning those building blocks into new materials like higher value materials like maybe Kevlar or something fancier than just a plastic bottle. And then there is another part of the team that's focused on this infinitely recyclable idea where you can just loop over and over and make polymers and building blocks back and forth. So Lannel is focused in these areas. So it's the bioscience division at LANL that's participating in this project. And so all of our work is really focused on using biological solutions to tackle some of these challenges. So under deconstruction, we're focused on using biocatalysis and bio-based organisms, microbes, to do the degradation. And then once we have those building blocks, we also have a part where we want to use microbes and collaborate with other folks on the team to help make some of these upcycled materials. And there was a really nice article in LANL's 1663 journal that came out a few months back. And the person who pulled that article together, Rebecca, came up with this great idea of saying, well, not microplastics, but microbe plastics, which I thought was a really, really great term. And I think we're going to post the link to find that article if anyone wants to read more. It's a really accessible and great article. So we want to use biology to degrade plastic. Well, is that even possible? It turns out that it is possible, and it's been identified that there are certain microbes that were found out in the environment that can actually degrade plastics and then even eat those degradation products. And so this is just a little cartoon of these microbes eating the plastics. But the way it works is that you have a bacterial cell, and the bacteria secrete a type of protein called an enzyme. And enzymes are able to do chemical reactions, and they're able to do chemical reactions faster than if those reactions were just happening on their own. And so the bacteria secretes these tiny proteins. The proteins stick to the plastic and start breaking off pieces of the bottle or of the plastic. And then those tiny degradation products, then the bacteria can actually eat those and use those as a food. So this was discovered these particular enzymes were discovered about 5 or so years ago, and that was really exciting. But the challenge is that if we want to use these enzymes to actually drive a process of degrading lots of plastic, the enzymes just by themselves found naturally, their activity is too slow. And it also turns out that trying to improve those enzymes or make those microbes work faster and better is also a slow process. In the laboratory, a lot of times we make one change at a time, or maybe a small handful of changes at a time. And then testing how well each change has given us the next step of progress is slow. And so this is where Los Alamos comes in to try to speed things up. So we have a technology that is really pioneered by Dr. Ramesh Jha, who's hiding back here behind this glowing green petri dish. And the technology is called Smart Microbial Cell Technology. And this is it's basically a suite of approaches that allows us to really fast track the discovery process for finding new and faster enzymes and new and better microbes. And this cartoon shows how it works. So the idea is that you take up some kind of nutrient or molecule from the environment, carbon source in this particular case, that an enzyme, so one of those types of proteins, chemically turns that molecule into something else, maybe a product that we care about. Maybe it's a monomer that we can then turn into a new plastic in the future. And so that target molecule then binds to this other type of protein. And this other type of protein is called a transcription factor. And what Ramesh does is he is able to customize these transcription factors to recognize and bind only to these target molecules and doesn't bind to anything else well. So when there's a presence of this target molecule, the transcription factor interacts with it, and then what it does is it turns on a third protein, which causes the cell to glow green, called green fluorescent protein. And so now the more target molecule you're making, the more your cell glows green, and you get in this case, like this picture here, we have a lot of different groupings of cells, and all of them are glowing this bright green. So now you can really see visually when your cell is doing what you want it to do. So instead of making a single change at a time or testing 10 or 100 maybe even different cells in a given day, now we can screen thousands of variants, thousands of different enzyme changes in a single tube. And this is just an example of how we've done that previously. This was published last year. And so in this example, we were starting with a microbe that makes a lot of a particular product that we cared about, but it actually grew really poorly. So the more we engineered the microbe to make this target molecule that we wanted, the worse it grew, the more unhappy it became. And so we want to try to regain the growth while keeping that, that production of our target molecule. So in this particular story, we had this tube shown here with these red cells inside, and these were our high product levels but low growth, so calling that number 1. And then we grew those over and over again in new test tubes until they evolved and developed mutations that allowed them to grow better. And this is called adaptive laboratory evolution. It's a really pretty common approach for getting your certain cells to grow better. And then what we ended up with, though, was we had cells that grew better, but now we also had cells that didn't make any product at all, or at least not very much. And so we didn't want that. We needed to keep our target molecule levels high. So what we did was we added our transcription factor, our sensor here, which I'm showing as this kind of glowing bright symbol, and we inserted that into every single one of the cells in the tube. And so now we are able to see some cells inside this tube glowed really brightly, and some of them glowed really poorly. And using an approach called fluorescence activated cell sorting, we were able to pull out or literally isolate the cells that were the brightest green. And when we do that, we actually get back samples that now have higher productivity than number 1 or number 2, either our starting population or the faster growing population. And not only do they have a higher productivity, but they also have get to their highest productivity in about half the time as the other cells did, or as the other test tubes did. So this really shows that we can make a lot of changes at once, and that we can pretty easily, like in one experiment, pull out cell types that are improved for the productivity that we care about. So how would we apply this to plastic degradation? Because what I just shared with you is really about synthesizing or making new molecules. And what we're talking about in the bottle project is degradation. Well, this little graphic here shows how we can adapt our smart microbial cell technology to plastics. And so here, the idea is that our enzymes, varying enzymes that might degrade our bottle, are in a cell. Each cell has a different type of enzyme. The enzyme is secreted outside to interact with your plastics. It, again, kind of chews up the plastics and creates these degradation products. And then the degradation products are taken up by the cell. And then now we've created new transcription factors or new biosensors that bind to those degradation products. And so only when the enzyme does a really good job of making a lot of degradation products do we get a lot of our green fluorescent protein. And so now we can look for cells that glow green, and those should be the ones that are doing the best job at degrading our bottle. And because bottles are solids, we can't exactly do this very easily in a test tube. We have some kind of substitute assays for that. But what we can do is we can grind up the bottle until it's in very, very tiny, like a powder. And then you can suspend your powder in agar in a Petri dish. So just basically a gel in the Petri dish, like you might have seen when you grow bacteria in, a biology class. And then you can actually look at your different cells, just like Ramesh was holding up in front of his face in that other slide. And now only some of these colonies or groupings of cells are going to glow bright green, and those are the ones that are doing the best job of degrading that plastic powder that's inside the agar. And so that's the concept that we are applying in bottle in order to be able to apply our technology to plastic degradation. And so this project is actually still pretty new, and so we're still in the process of really ramping up our tool demonstration and kind of proving that this can work. But we've made some really great progress. And so first of all, here's just the same graphic that I showed from just the slide before. And what we've started first is just trying to develop these transcription factors. So like I said, what we do is we have to customize the biosensor to bind only the degradation product that we care about and nothing else, so that the signal only turns on when degradation is happening. And so this is an example of showing that we've gotten that to work for a particular degradation product called terephthalic acid. And so this just shows that if you have no terephthalic acid present, the signal of fluorescence is low, and then when you add terephthalic acid, this signal becomes brighter. And so we know that the transcription factor is turning on in the presence of terephthalic acid. So that's one piece of this this technology that we would use. And then also I showed you this cartoon idea of being able to pick bright colonies off of a petri dish, but does that really work in real life? Well, it turns out that it does, and we've shown that actually in a previous paper, and that's shown here. It's a little bit grainy, but you can pretty clearly see, I think, that there's a lot of cells here that are dim, and they're not turning on that green fluorescent protein very brightly. And then there's this one cell that's glowing very brightly in the center, and that is bright enough and big enough that we can literally pick it up with a toothpick and analyze it further. So we're currently using these technologies, as well as some other technologies that I didn't have time to share today, to start really screening for better degrading enzymes. And beyond your standard, water or Coke bottle, we have a number of other polymers where we're starting to think about how we can have some impact as well. And so those are shown here. So we're really interested in nylons. Nylons have a type of polymer structure that should be degradable using biology. Some harder problems are polyurethane. But these types of foams are everywhere and hard to recycle, and so we are interested in working in that space. And then you might be aware that there's a lot of products that actually have layers of different polymers, especially certain packaging for food and other types of packaging have multiple layers of different polymers to make a single plastic, looks like a single plastic. And so each of these would require a different biological solution in theory to degrade, but then you can mix and match your varying enzymes that you've developed into a single cocktail to degrade something like this. So that's where we aim to go in the future. And then for the upcycling piece, that kind of second side of the area that we're working in past deconstruction or degradation is this upcycling and infinite recycling idea. And so as I mentioned, part of the team is designing new building blocks for more valuable or more recyclable plastics to get at this circular plastics economy. Part of our team in Bottle is looking to find new organisms that will eat all these different types of products that would come out of degrading a lot of different polymers, our 1s through our 7s. And the idea is that if you can find an organism and then engineer an organism to take up all those different types of various products, then you could get it, in theory, to turn them all into a single type of building block that you could use to make new polymers. And so our role in this would be similar as to what I've already described, which is that we would build new sensors for these new building blocks that have been invented to do the circular economy. And then we can use those sensors in the same way that I already explained. So here I have written target molecule, but if you substitute target molecule for building blocks, then this concept is identical. It's just that now you've made, again, a different sensor that's specifically set up to check out those building blocks or to screen for cells that make those building blocks the best. So this is the end of my formal part of my talk. I wanted to acknowledge all the bottle team members. I couldn't possibly list all the names that are part of this project. So for a number of the institutions, I've just listed the lead. In cases where I've listed more names, it's because I've actually talked about work that they've been working on, or they're active members of the Lannel Bottle Project. And so I think I'll leave it there and acknowledge again that this is funded by the DOE, and I'm really pleased to be here and can't wait to answer questions.
Taraka, this is just great. I didn't interrupt you when you were at the point of talking about the fluorescence work, but I'm struck that you've leveraged some previous discoveries at Los Alamos National Laboratory in this work, the fluorescence labeling work, right?
Yeah, the fluorescence activated cell sorting. So fluorescence activated cell sorting was invented here at Los Alamos, and the use of flow cytometry and the development of new applications for using flow cytometry across a really wide range of problems is something that Lannel is really known for. And so, yes, that's one of the reasons why we were brought into this, is because of that piece of it.
Would you just briefly define for the folks out there how does flow cytometry work and what is the theory behind it? Because it's developed here, but it's used everywhere.
Yeah, so I will hope I can do this as much justice as I should be able to do. I usually have like a cartoon of this, but the idea behind fluorescence activated cell sorting is that, first of all, just flow cytometry analyzes a single cell at a time and detects properties of that cell based off of light scatter. So if basically part of the process is that your sample coming from your test tube has, it causes your cells to line up single file, and then a single cell reaches a point where it's interrogated by a light source, and so a laser will hit your cell, and the light will bounce off of the cell. And so that can tell you things about how large the cell is or how many particles are inside the cell. And then another piece of it, though, is that if you have something inside your cell that is fluorescent or can be excited to be fluorescent, like green fluorescent protein, when you hit the cell with a certain type or certain wavelength of laser, then it will glow green. And the more of that protein that's in there, the brighter green it will glow. So that's how it kind of turns on the glowing. And then the sorting piece is a little bit different. So the sorting piece has basically a sensor where once the cell meets a certain criteria based off of the fluorescence or the light scatter, then the instrument uses an electrical pulse to send your cell into a certain tube. So basically, then you can bin your cells into new tubes based off of how they basically how bright they are. Yeah.
That's just such a great technology for so many applications. And it just reminds me that we're known here at Lannel, Los Alamos National Laboratory, for sensors. We develop great sensors to be able to detect things.
Yeah.
And the uses, it just takes a need, and someone will say, that thing that we developed, it could be used toward this other thing. And that's one of the strengths of our lab, is that we've got so many different projects going on in so many different areas of expertise, and they can come together and come up with a great idea for a new approach, which is, it's the discovery process that you're really talking about here. That's just so fantastic.
Yeah, that's true. And so in commercial flow cytometry, a lot of what is used is they have special dyes that you can put into the cells and cause them to light up in a certain ways, certain colors, fluoresce certain colors, based off of various properties. And so in some of our other projects, like the algae project, we're able to use some of those commercial dyes to tell us if a cell is making a lot of oil, and therefore it might be good for biofuels. And then we use the cell sorting concept in just the same way. And here, because these target molecules are so special, we don't have a commercial dye that we can just buy to detect them. And so that's what's so special about the transcription factor-based biosensors, is that there we're actually using a totally different capability that's been developed and recognized here, and that's computational protein engineering. And so Ramesh uses computation to decide what changes to make to try to customize that sensor to work for a certain molecule. And so that's a really great example, actually, of combining multiple capabilities that have been developed at Lanel.
So I want you to explain what I think I already understand, but there are some interesting questions here. So Jason wonders, what kind of environmental growing conditions do you use for the microbes to do their work? And here's his question too: have you tried to mimic conditions in a landfill or industrial conditions? So it's really, the question is, so where is this going to take place?
Yeah. So in the lab, we just grow them in a regular test tube under kind of pretty warm conditions, like 30 degrees centigrade. But in practice, what we envision, at least the way I envision it, is that we would be using, large tanks that are enclosed, that have our engineered microbes in them, and then you would basically feed your plastic to those tanks to be degraded, and then they would the same microbes would use that degradation product to turn into a target molecule that we're interested in, and then we would kind of harvest that molecule to make the next polymer.
So it basically becomes like way downstream from this wonderful baseline work that you're doing. It's a factory. It's a recycling facility where they'd have the microbes in tanks. You'd put in your stuff to be degraded, to be upcycled or broken down, so that you've got the components, and you harvest that from there. And those plants probably could be built where you have recycling facilities, or maybe built close to some of these terrible plastic islands that are out there, right?
Right.
Yeah.
Right. Yep.
Yeah.
That's right.
Oh, okay. And along with this too, Jerry asks, what is the fate of plastic-eating enzymes? Are you creating a Frankenstein that will be turned loose in the environment?
Yeah, I don't think so. I mean, I get this question pretty often. The idea is really, as you just described, Linda, where we would be using these in a carefully controlled and closed system. I, again, envision this in using the same types of fermentation tanks that are used in pharmaceuticals and things like that. And, the types of quality control in those areas of work are really tight. And so we'd be using the same types of rules and same types of quality control to maintain our microbes just where we want them.
And you'd want that because you're looking for really great building blocks to come out of that process, so you can upcycle it.
Right.
And do those great things with it.
Right.
Right. Let's see. You showed so many partners, and that's another wonderful thing. Not only is it like across the laboratory with different kinds of competencies, but there's so many partners that are involved. And we got a question from Kathy. Have the other research institutions made as much progress as Los Alamos National Laboratory on their portions of the project?
They have, and I would say we're actually a handful, if not maybe a handful, it might be fair to say, not a handful, about 4 of the organizations were already working together, 4 or 5. I don't want to misspeak, but I also don't want to count in front of everyone. A handful of folks were already working together on some other work that was funded in this area, and so they already actually have more papers and amazing progress. So we're coming in sort of, I wouldn't say anywhere close to the tail end of it, but there was already some momentum, and they were actually getting to the point where there's some great papers, and you can find them on thebottle.org, of some improvements that they've already made in the enzymes, but now they're just getting to the point where we really want to screen faster, and that's really our strength.
So all the partners that are on this slide, are they specifically on the microbe part of it? Because one of your earlier slides, you showed 4 different approaches that you can take to getting the components for upcycling. But these are the partners that are specifically on the microbe part.
No, these are the partners that take
across
care
it.
of the whole thing.
Okay.
So when I showed the whole thing and said we're doing, parallel approaches, these are all the partners for that. And so MIT is very focused on some of the other degradation approaches. And so actually Yuri and I co-lead the deconstruction task. And NREL has a big part in some of the chemical deconstruction, but also the biological piece, as well as we do a lot of techno-economic studies and that, sort of thing. And so they're involved in that. And then a number of them are biological. So Montana State is mostly focused on biology. University of Portsmouth is very focused on the biology. But Northwestern is more focused on coming up with new ideas for coming up with the redesign, so new molecules that we might want to shoot for. And the whole Colorado State is actually leading the redesign effort. And so Eugene has got some amazing approaches for coming up with new polymers. And so, with just completely unexpected monomers or building blocks. So it's everybody. This is everybody. These aren't all the names, but these are the institutions. Yeah.
So since you mentioned that, let me ask this. What do the degradation molecules look like? This is from Amy. Are they the basic monomers, or are they short polymers?
It depends. So if you get, at least in the case of PET, which is what I was mostly talking about today, there's 2 primary degradation products, and they are, they're the monomers. But enzymes often degrade things into kind of chunks, and then there's other enzymes that degrade them further, and that's the case here. So you do also get kind of mini polymers, if you will.
Well, and if they're useful, if the mini polymers are useful, then you're good to go.
Yeah,
and you can do something with them.
Yeah. And then there's additional work to kind of, there's this second enzyme that, again, didn't have time to talk about it at all today, and so there's already work trying to improve that as well to get everything down to those final building blocks. But there are other polymers that do not degrade into their original monomers. So just the way the breakdown happens is you get something slightly different than what you started with.
Yeah, and that must be a fascinating part of your discovery process now, of seeing what those variants can do for you. You know that they're breaking it down because they're bright green and they've produced, but what are they really good at, and what could we leverage? This is a really great question from Steve. What are the byproducts of the microbe enzymes? Does this process release CO2 from the plastics, which would then not be a great contribution to climate change?
Yeah, that's a good question. To my knowledge, CO2 is not a direct breakdown product of these plastics. But the breakdown products do have carbon in them.
Yeah.
And so we want to direct as much of that carbon into making these new building blocks. And at least in the case of the biological approach, cells do respire and give off CO2 as
Right.
part of their metabolic process. So part of the engineering is to take that into account and try to make sure that we're making more of the building block than we are of just making more
Yeah.
CO2. But, again, if it's in a controlled system, the way there are various other technologies that utilize CO2, you could imagine potentially coupling this to something that would collect that CO2 versus just blowing it out of a smokestack. So,
Right.
there may be ways to align this with other technologies. And I mentioned we're doing techno-economic analyses, but we're also, and the Bottle team, are also doing life cycle analyses, and they take all of that into consideration. So how much heat and energy do we use? How much water does it take to grow these things? Because you have to put them in basically water with a bunch of nutrients, a media. How much CO2 does it make? And if it doesn't make sense, if you're doing worse than doing nothing, then that's a no-go for
Right.
us. Yeah.
Right. It's not going to go anywhere. There was another one. Let's see. Carol comments, what important work. We hope funding sources are aware of your work in progress and are going to continue to assure your funding. So what does that outlook look like? We know that you're being funded. The Department of Energy is so interested in this. What other kinds of streams do you think are out there to push this forward and actually make it into, technology transfer, which is something we do here at the lab? We develop a great way to do something, and then it gets let out basically into the open for businesses to get really good at doing that and take it on.
Right. Yeah. So we're kind of just getting started. So we expect to be funded for quite some time. There's the U.S. has put out a number of documents. One of them is the Plastics Innovation Challenge, where DC has really taken on this problem and is really interested in this currently. But in terms of tech transfer, I mentioned that companies are interested in this problem, and we have, from the start of the project, and even in our kind of like project development year, have been on the outreach campaign for connecting with companies and, talking with them about what they think would be really useful, so that we could ideally hand off as much of our technology as possible to people who are actually making and using these packages to get their products delivered. And so I can't really speak about which companies are seeming really interested, but we have had some success there. So I'm hoping that we're going to be able to, announce at some point that we have some actual company partnerships where they're asking us to do certain R&D projects for them with their specific needs in mind.
Because the cell, the microbe, is very targeted to do a really great job on a particular kind of feedstock. Mm-hmm. Does this mean that we're going to continue to sort our plastic into the future, because that'd be a great way to ensure that you get the purest feedstock for a particular kind of microbe that's doing a great job? Or is sorting, is that another nut to crack, where, it's just put all your plastics here and we'll figure it out?
Right. We would really like to end up with this model that I'm showing here in the middle, which is that whether it's different enzymes all mixed together, or whether it's a chemical process that is able to take a bunch of different plastics and just melt isn't the right word, but just degrade everything and then have all those different degradation products. But we would like to make a soup so that people don't have to do as much sorting. And then engineer our microbes to eat that degradation product soup and then turn it into, we might have one tank that then takes that soup and turns it into one building block so that it's easier to harvest, as you say, that building block, and a different tank maybe that makes a different building block. But the soup, ideally, is the way to go. We aren't actively working on the sorting side of things, but the Advanced Manufacturing Office, which is one of our co-funders, co-sponsors, does have other projects in that area. And so, there's kind of an awareness of what everybody's doing and, an idea of trying to coordinate as needed in the future.
Sort of corollary to this, in your approach, this is from George, in your approach, you select for a cell that has maximum performance. In some processes, it can be a consortium, perhaps, that's symbiotic in achieving a final goal. Can you sort for complementary cells that work together?
Yeah, that's a super fun question. So, there are different fluorescent proteins, and so you can tie this type of system to, say, a red fluorescent protein for one target molecule or one degradation product, and then green fluorescent to another, and then you can use this same type of idea on a two-color, or maybe I'm a little reluctant to say multicolor. That might be a little beyond what we can do in the very near term, but you can use dual to maybe even 3 colors. And so you could, in theory, mix your cells together if each one was reporting on a different color and try to pull out ones that, and you can also put, those same pathways in a single cell. So you could have a single cell that makes both red and green. And if they're both working really well, your cell will actually appear yellow in the flow cytometer. And you can, that's definitely been shown that sort of thing can be done.
Oh, that's fascinating. That's fascinating. And so when we're talking about the microbe, I don't know what, originally I was thinking algae, and then I went on to think about bacteria. Is it both? Is it neither?
It's bacteria.
It's bacteria.
Yeah, so this particular project and these related projects with the smart microbial cell technology are bacterial. So we would love to use this in algae, but the tools for engineering algae and putting this type of technology into algae cells, at least the types of algae that we work with, is not there yet. So the foundational understanding of most of the algae strains that we work with still require a lot more work before we can use something that's kind of fancy.
Yeah.
So that's why we just use the commercial dyes. And even that, even just tailoring the protocol to get one of those to work, where they're usually designed for human cells or bacteria, and then you're just trying to adapt it to get it to work in algae can be tricky. So, yeah, one day, hopefully.
Yeah. Well, you're doing very intriguing things with algae as well, with for biofuels and optimized lipid production, so that grow your own jet fuel. Right? Yeah. Interesting. From Donovan, is the bottle project also looking at life cycle of bio-based polymers?
I might. It's a little hard to know what he means by bio-based polymer. So if he means a polymer that a cell is actually, a polymer that a cell actually makes inside the cell versus making a building block and then harvesting it and converting it into a polymer outside of the cell. The current building blocks that we are actively talking about in bottle are not bio-based polymers. But we do have other projects at Lannel, and there are other projects out there, plenty out there in the world that look at bio-based products. But we have 2 really great projects that are funded by the LDRD, the Laboratory Research and Development oh my gosh, organization within the lab.
Yeah.
And so we have one that's focused on polyhydroxyalkanoids, which is or PHAs, which are really well known, actually, bio-based polymer for packagings, and that's run by Dr. Babs Maroney. And then we have another project that I co-lead with another person that's focused on making bio-based polymers that can be used in things like diapers and coatings and adhesives. So they're adsorbents. And so they're a really different type of polymer, but we've also probably all heard those stories about all those disposable diapers that are in the landfill and they're going nowhere. And so I'm pretty excited about that particular project. But in Bottle itself, we're focused on these types of building blocks that are not made into polymers inside the cell.
Well, Don asked the question, how many bottles can you at this time break down per day? And it seems to me that, this is the starting stage.
Yeah.
You are developing the tools to be able to begin working with these microbes that are going to do their good work, But let me ask you this variant of that question. How far off do you think we are from seeing a real practical application of this that we're going to start employing it?
Yeah. It is a really hard question to answer. You're right that we're still in the very early stages of R&D. I think I mentioned that, the first enzyme of this type was just discovered 5 years ago. It took 2 more years to make one better variant of that enzyme. And then it's taken two more years to have another publication out that shows you can kind of use this 2 enzyme system in a special way. So it does take time. I'm hopeful that, especially with the kind of high throughput approaches that we're bringing to the table, that it'll go faster. The goals for Bottle, I think, are, laid out at the, we usually tend to think of our kind of big goals at the kind of 10-year mark. Mm-hmm. But how fast this will be really picked up is pretty hard to predict. We aren't reliant only on companies to help us figure out the scale up, though, for example. So NREL has got scale up facilities and really these huge tanks. And so there will be opportunities if we get something good to practice and have a feel for how it works at scale, at a larger scale, as soon as we have something decent.
Right.
So sorry to not truly answer that question. I hate doing that, but it's difficult to say.
Well, but that's the future,
Yeah.
right? And we'll see what happens. But, I think what we all know is if there's money to be made from it and really something better to be made from it, and we all know that unfortunately we have such a supply of plastics already. It's like that resource is out there. It's begging to be used.
Yeah, we had talked about this in one of our chats earlier. The idea of actually mining plastic, the idea of really using plastic as a resource, a carbon resource that's out there, I think is a really interesting way to think about this. And not only is, if there is money to be made because you can, mine that and use it to make something else, but also, the more people care about it and the more consumers care about it, that does apply pressure, for lack of a better word.
Yeah.
And that means that we'll get that much more support to keep working on it. So.
Well, I can't imagine that there's anybody out there that's not rooting for you all to keep making progress and for the Bottle Project to be a huge success.
Thanks. I hope so.
Yeah. Thank you, Taraka, so very much. This has been a wonderful conversation. And thank you all out there so much for your great questions. It really explored the topic very well. So this is the end of this month's Science on Tap. We look forward to seeing you next month as well. Thank you so much. Bye-bye.
Thank you.